presented just now, a fantastic in-depth application of flow cytometry in one aspect of bone marrow investigations, whereas I'll be presenting the general basic principles of flow cytometry and its use in hematological malignancy as well as its role in minimal residual disease assessment. So what is flow cytometry? So it is basically a study of cells in liquid suspension. It can study basically all particles as long as they are suspended in liquids and as long as these particles have any target antigens. So we need a flow cytometer of which the main components are fluidics, lasers, optics, and a software for data acquisition. And the main reagents for flow cytometry is fluorochrome conjugated monoclonal antibody. So the fluidics of the flow cytometer will transport the cells of the sample to the point of interrogation by the lasers. And at this point, mainly two things happen. The light energy from the laser is scattered just simply because of the nature of the cells, and these scattered light will be captured as forward and side light scatter characteristics of the cells. And the second thing that can happen is that if the cells have attached fluorochromes on the surface or inside the cells, these fluorochromes will be excited by the laser light, and light energy again will be emitted from these fluorochromes, which will be captured by the relevant optics of the instrument, which again is amplified in the photomultiplier tubes, and finally this signal will be converted into electronic data, which we will see and analyze later on. So the first important step in flow cytometry is to set up the instrument voltages for forward and light side scatter characteristics, as well as for individual fluorescent channels. Of course, a fluorochrome compensation setup has to be done as well to correct the spillover effect of fluorochromes into adjacent channels, and daily QC runs are important for all these settings to be stable over time. So this is an example dot plot of a blood sample just based on the side scatter and forward scatter characteristics. So it is easily a recognizable pattern when we are dealing with blood samples if we are familiar with it and if we are doing it in day-to-day basis. So here we can see that the small particles, small cells, which are mainly lymphocytes and some small blasts, will be located here. And the medium-sized cells, mainly monocytes or medium-sized blasts, will be located in the middle region, with the larger cells being neutrophils or eosinophils will be located in this region. So this is all about pattern recognition, like Dr. Lee has presented just now. Okay, so now we'll move on to the role of flow cytometry in hematological malignancies. So basically, it is a very rapid assay which can be performed in a matter of hours, and we can add as many parameters as we want to interrogate a sample by flow cytometry for the immunophenotyping at diagnosis, as well as minimal residual disease assessment. Other uses of flow cytometry include cell enumeration like CD34 quantification or lymphocyte subsets quantification, cell viability analysis, cell cycle analysis, and cell sorting. And it is also useful for non-malignant hematological conditions such as congenital platelet disorders or paroxysmal nocturnal hemoglobinuria or red cell membrane disorders like hereditary spherocytosis. So for hematological malignancies, immunophenotyping is very important for the diagnosis as well as for prognostication. For example, almost all the acute leukemias are basically diagnosed mainly by immunophenotyping. And by knowing the immunophenotyping, it sometimes enables us to predict the underlying cytogenetic abnormalities or associated molecular abnormalities, because some of the immunophenotypic abnormalities are found to be associated with certain genetic mutations. So to perform immunophenotyping by flow cytometry, we need to first design multicolor panels with fluorochrome conjugated monoclonal antibodies after selecting the target antigens on the specific cells of interest. And these multicolor panels have to be validated and improved as necessary. And the laboratory has to develop standard operation protocols for sample preparation and sample acquisition. And the test sensitivity has also to be determined by the individual laboratory. And finally, data analysis and interpretation can be done by a trained laboratory staff or by the trained hematopathologist. So So these are some basic rules that we follow to design multicolor panels. Basically, since the samples that we are dealing with most of the time are hemopoietic samples, so CD45, which is common leukocyte antigen, is added in almost all the tubes, and then backbone markers are selected to identify different lineages of the cells. And subsequently, relevant target antigens will be selected based on the published evidence or based on the experience of the laboratory. And when we design multicolor panels, we also need to consider the type of specimen that we are dealing with. One common rule for the choice of fluorochromes is that brighter fluorochromes are usually used for investigation of weakly expressed antigens or the antigens of unknown expression. And of course, we also have to consider the spillover effect of a bright fluorochrome into neighboring channels. And these are some examples of target antigens found on various types of hemopoietic cells. For example, if we want to investigate the stem cells, we can select antigens like CD34, CD117, CD33, CD99, and so on. Okay, so this is just one example of multicolor panels for the diagnosis of hematolymphoid malignancies, which can be found on Euroflow Consortium website, and they are basically Euroflow panels. And in the laboratory that I work, we had 5 color panels, as well as we have Euroflow 8 color panels. So this is an example for the diagnosis of B acute lymphoblastic leukemia. So here we use CD45, CD34, and CD19 as backbone markers to identify the B lineage cells, and other target antigens are added based on the B cell maturation as well as to detect the aberrant antigenic expression. Similarly, for T acute lymphoblastic leukemia, cytoplasmic CD3 and surface CD3 together with CD45 are added as backbone markers, and the rest of the antigens are based on T cell maturation as well as aberrant antigenic expression. And the same goes for acute myeloid leukemia. So at one look, we may think that the panels are too extensive, but then again, to diagnose acute myeloid leukemia, if we look carefully, the first tube is basically to study the maturation of myeloid cells and the myeloblast, and the second tube is basically to study the monocytic differentiation, which included CD64, CD14, CD35, and so on. So basically, most of the acute myeloid leukemia will fall into the category of myeloblastic or myelomonocytic differentiation. After running these 2 tubes, most of the time we will be able to diagnose acute myeloid leukemia, and only in rare instances we need to expand the panels to include panels for erythroleukemia or megakaryocytic differentiation. And when we interpret the data, again, it is important to look out for internal controls. And it is very easy for us because, well, the samples that we are dealing with is mainly hemopoietic samples, so we always have, almost always have normal hemopoietic populations, other normal populations. And most of the time, they are normal lymphocytes, which we can use as negative controls. And isotype controls are nowadays almost obsolete in most of the laboratories. And rarely, we may need to use fluorescence -1 controls, especially if we want to check for artifacts or we want to know the position of negative populations. So when it comes to data interpretation, it is all about pattern recognition. Dr. Lee has actually emphasized many times in his presentation about comparing the normal pattern against the abnormal pattern. So, so basically, what is abnormal pattern? So abnormal patterns means recognizing abnormal size, abnormal quantity, absence of normal antigens, or presence of abnormal antigens, or presence of normal antigens with abnormal intensity. And again, we cannot assume that neoplastic cells will be where normal counterparts are. So here it will be useful to use data interpretation tools like flow cytometry data analysis softwares. So this is an example of pattern recognition. So on the left is the dot plot based on size scatter and CD45 expression of a normal blood sample, and on the right is an abnormal blood sample. And instantly we can see that there is an expanded population in the CD45 dim area and the low to moderate size scatter area. So now we'll move on to the last part of my presentation, which is the role of flow cytometry in MRD assessment. So an MRD assessment tool should be sensitive, accurate, At the same time, it should be feasible for almost all the laboratories and clinically relevant. So these are the currently available different MRD tools, including flow cytometry, real-time quantitative PCRs, and high-throughput sequencing, like next-generation sequencing. And the sensitivity of all these tests are, in general, comparable, especially with the introduction of next-generation flow by the Euroflow group, with the test sensitivity reaching up to about 10 to the power minus 5 to 10 to the power minus 6. And of course, each individual test have its own advantages and disadvantages. So which tool to use in your laboratory has to be decided based on the feasibility and availability of expertise and facilities. For MRD assessment by flow cytometry, usually there are two approaches. First is to look for leukemia-associated immunophenotypes, and second is to look for difference from normal immunophenotype. But in practice, most of the time, we combine these two methods when we look for MRD. So leukemia-associated immunophenotypes include cross-lineage antigen expression, for example, in acute myeloid leukemia, if there are presence of lymphoid antigens, or asynchronous antigen expression, which means the presence of mature and immature antigen expression simultaneously, or over or under expression of normal antigens. Whereas for difference from normal immunophenotype approach, we need the knowledge of normal differentiation and maturation of hemopoietic progenitors. And at the same time, we also need to have the knowledge of therapy-related changes to identify a population whether it belongs to the abnormal or normal group. This is just an example of MRD panel for B acute lymphoblastic leukemia by Euroflow group. So here we can see there are 8 fluorescent channels used, and 7 channels are occupied by the backbone markers and B maturation markers. And this Euroflow MRD panel is successfully reproduced in our laboratory with almost similar sensitivity, where abnormal B lymphoblastic leukemia cells are identified at a very low level. So what is the clinical relevance of MRD? So MRD status is proven to predict outcomes for both chemotherapy and hemopoietic stem cell transplantation. It is also reasonable to modify treatment based on MRD status in some diseases. It can also be used as a surrogate marker for outcome assessment. And in a clinical trial setting, if MRD can be used as an endpoint, rather than the conventional endpoints like event-free survival or overall survival, the new therapeutics can be made available faster for the clinical practice. And the use of MRD for the long-term surveillance of diseases is still under evaluation. So this is just to prove that MRD in acute lymphoblastic leukemia is a strong independent risk factor to predict event-free survival and overall survival for both pediatric and adult acute lymphoblastic leukemia population. And as for multiple myeloma, MRD status is now incorporated into the response criteria assessment, so by IMWG group. So this MRD can be assessed by flow cytometry as well as sequencing techniques. So this is the overview of clinical relevance of MRD for different diseases. So for pediatric ALL, it is proven to be an independent risk factor for disease-free survival, overall survival, and also prediction of relapse risk. Risk-adapted therapies are also proven to change the outcomes in pediatric ALL population. So risk-adapted therapy means can be both ways: intensification of treatment in MRD positive patients, or de-escalation of treatment in patients with negative MRD. For adult ALL, the outcomes are not as good as pediatric ALL, so it can be a bit challenging to de-escalate the therapy based on MRD status. However, there are some studies showing that the results are promising by intensifying the treatment with the incorporation of stem cell transplant for patients with positive MRD. And the same goes for AML. MRD status is relevant for a prediction of disease-free survival, overall survival, and relapse risk. However, based on MRD status and to modify treatment is still complicated in acute myeloid leukemia, because in some of the acute leukemias, persistence of some genetic mutations does not really lead to clinical relapse. So if we want to base on MRD status to modify treatment in acute myeloid leukemia, it has to be done at physician discretion or in the setting of clinical trial. And as for multiple myeloma, it is proven to predict disease-free survival and overall survival. But to initiate a therapy based on MRD status or to change therapy based on MRD status is still unknown at the moment. And some effort has been put in to correlate the different MRD tools. So high concordant rate was found between the MRD tool by PCR method, MRD assessment by PCR method, and flow cytometry method. However, if we compare the flow cytometry method and high throughput sequencing method, the results are a bit variable depending on the different laboratories where the sensitivity of the flow cytometry tests are variable. Okay, so in summary, flow cytometry is an essential tool in the diagnosis of hematological malignancies. Multicolor panels can be designed based on the published evidence and the experience of the laboratory. And MRD assessment in hematological malignancies is a rapidly developing area, and flow cytometry can be successfully applied using MRD protocols. Thank you very much.